Good morning, and good morning to those people that are listening to us through live stream. My name is Professor Sonia Marshall-Gradisnik. I'm the co-director of the National Centre for Neuroimmunology and Emerging Diseases. I'd like to thank the organisers for inviting myself and my colleague, Professor Staines, to present at this conference. And it is with my greatest delight I get to introduce the first speaker for this session. The session will have two speakers, and Professor Staines will introduce the second speaker. And may I just remind those people that are presenting in this session, the session has a time frame. Professor Fisher, I'll give you a bell within the minute of finishing, 45 minutes, and that will allow then question time for the audience for 5 minutes. If there is any questions. So it is my delight to introduce Professor Paul Fisher from the Department of Physiology, Anatomy and Microbiology at LaTrobe University. Paul's area is obviously neurodegenerative diseases and mitochondrial biology. I welcome Paul to present, and I'm most interested in looking at your research, Paul. Thank you.
Better see if I can use this machine first. At my age, you get a bit technologically illiterate sometimes. So, first of all, I want to say thank you to the organisers for such a great job for organising the conference, and I'm honoured to be here. I'm honoured to be presenting in front of fellow scientists who work in the area, some of them whose names are really well known to me and whom I've not met before, and also to be presenting in a live stream and also here in the room to patients and carers of patients, family members and so on, who I've learned in the course of working on this are incredibly intelligent, incredibly diligent in researching their own disease, and they know an awful lot about the disease, which makes it really daunting talking to them because they probably know more than I do. So, as said, I've been working on mitochondrial biology for about 30 years now, and I spent some time talking about neurodegenerative diseases, researching those. And I started to get into chronic fatigue syndrome research or tried to get into it about 10 years ago, but it took me quite a while to find a link that would allow me to get access to patient samples. We finally did, and the people whose names you see up there are the people whom I've collaborated with in this work. I've highlighted two people in particular, Daniel Misalidis, who is a PhD student in my lab and has done most of the experiments I will tell you about, and Dr. Brett Lidbury, who was my first collaborator in the chronic fatigue syndrome area, and he's actually contributed his weighted standing time test for testing the severity of disease, and you'll see some results from that as we go through. In addition, Dr. Don Lewis, who runs a clinic in Donvale that gave us access to patients, Sarah Annesley, who's down here at the front, who co-leads my lab, and Juana Stanislav and Claire Allen, who are in my team as well. I want to say that the people in my team are a team of really fantastic, talented young scientists, and they're the ones who've done all this, and I just get to talk about it today. Okay, so let's move on. Enough of that. Oh, I should acknowledge also, our work was made possible by some very generous donations from a patient, and then from the Mason Foundation, and more recently from the McCusker Foundation, so I want to acknowledge them as well. I usually forget acknowledgements because I tend to forget things all the time. Okay, chronic fatigue syndrome is ME/CFS, myalgic encephalomyelitis, whatever you call it, is a big, complex problem, as everyone in this room knows, and I think it's a much bigger, more complicated problem than any of us can discern, which is why a conference like this is so important, because we can talk to each other and compare notes and try to figure out what's going on. So I wanted to present this to you. It's a representation of an old Hindi fable where 7 blind men, I think there's 7, in the fable it's men, in this image there's actually at least one female in there as well, who are asked to identify a creature, and they each get led out to the creature, and they each inspect a different part of the creature. One inspects the tusk and thinks it's a spear, one inspects the trunk and thinks it's a snake, and so on. the scientists in the room can try to figure out which of these they are. I like to think that I'm at the forefront, at the pointy end, but my family and perhaps my students might tell me I'm more likely to be at the rear end, bringing up the tail. But in any case, enough of the dad jokes. Let's move on. So, the question I want to address is the role of mitochondrial disease, mitochondrial dysfunction, let's say, in ME/CFS. Is it really impaired? So, if you look at the literature, you find that mitochondrial enzyme activities are reduced in ME/CFS muscles. The problem is that mitochondrial biogenesis and mass are upregulated by exercise. And of course, by the nature of the disease, there's less exercise in muscles from chronic fatigue patients. So you're compounding the problem of cause and effect, and it's really difficult to disentangle them. The second line of evidence is that when people have looked for direct measurements of mitochondrial respiratory activity in lymphocytes, they find generalised reduction of all respiratory activity. However, ex vivo lymphocytes are metabolically quiescent and dying. When you look into the literature, you see that people measuring the production in the proteome, in all the proteins in cells, they find that in lymphocytes and in platelets and in the saliva, numerous mitochondrial genes are upregulated, not downregulated. So what's the truth here? What's actually going on? To illustrate that metabolic quiescence, let me show you, jumping ahead of what I'm going to talk about. On the left-hand side, you can see that when you measure mitochondrial respiratory activity in immortalized lymphocytes, so these are ones that we have treated so that we can grow them in culture and store them and go back to them, you see consumption rates of oxygen, so mitochondrial activity, of the order of 400 or 500 picomoles per minute. But lymphocytes, the same number of lymphocytes under the same assay conditions, give you really, really tiny levels of rates of respiration. On the same scale, you can't even see it. If you amplify that scale up, you can see that you're talking about 2 to 10 picomoles per minute, so really low rates of respiration. And that causes problems then. Are you actually measuring the quiescence of the cells, or are you measuring the death rate of the cells? Because lymphocytes are also dying in culture. What are you actually measuring when you measure mitochondrial function in these cells? You'll get, I think, a really nice talk from Cara later showing you that in our experiments this difference here wasn't significant, but we only did a relatively small sample. Cara's going to show you with her really elegant work that this difference here in the lymphocytes is real, and you can see the CFS looks lower than the controls, but the question is, why is it lower? So I'm hoping to convince you that we've answered that question. Okay, so I've already touched on some of the reasons why we want to use lymphoblasts to study ME/CFS mitochondrial function. The first reason is they can be stored indefinitely. So you can go back to them again and again and regrow them in culture under the same conditions, or you can change the conditions and you can ask questions about, new questions about their mitochondrial function or other activities in the cells. Because they're grown under identical controlled conditions, regardless of the lifestyle, age, gender, hormone levels, blood, conditions, etc. of the patient, there should, in theory, be no difference, right, between chronic fatigue syndrome lymphoblasts and control lymphoblasts, unless there is actually something genuinely wrong with the chronic fatigue lymphocytes and therefore the lymphoblasts in the first place. So you know that you're not dealing with differences in the conditions under which the cells were collected. As I said, they provide an unlimited supply of healthy, metabolizing intact cells for larger scale repeater follow-up experiments. So if your experiment gives you a result that's interesting and suggests another hypothesis, you can then follow that up and go back to the same cells and ask that question. They are readily obtained from a single blood sample, and the results can be correlated with clinical data collected at the time of blood sampling. So we think that they're really useful. We have found them really useful in our work so far. So I'm cognizant of the fact that not everyone in the audience is a scientist. I'm cognizant of the fact that in the live stream, there'll be people watching who are not scientists. So I'm going to try to give you a bit of a crash course in mitochondrial biology in a way that will be accessible as possible with this mixed audience. So what we have here on the left is a cartoon of a cell with its nucleus where the chromosomes are and the mitochondria here. And this is one of the mitochondria blown up, and you'll see there's an inner membrane and an outer membrane, and between them is a membrane space. These little guys produce more than 90% of the cell's energy in the form of a molecule that you'll hear lots about called ATP. Over here on the right, we've got a real image of human cells in culture showing stained mitochondria, just so that you can see what the cartoon looks like compared to reality. Okay, so the fact that there's two membranes is important because a lot of the action in respiration and making energy happens in the inner membrane. So this is the inner membrane here. And what I want to do now is to show you how the process of respiration that we're measuring actually works and how we're actually able to measure it. When your body, when your cells, your mitochondria oxidize food molecules, what they're doing chemically is they're actually carrying out a reaction where electrons are passed from one molecule to another. The molecule that is being oxidized loses electrons, and the one that is taking those electrons, ultimately oxygen, we say it's reduced. The chemical jargon doesn't matter. The point is there is an electron flow. And this electron flow is happening through respiratory complexes, which I've just covered up for you inadvertently, because I wanted to make the point that these protein complexes that are doing all of this are actually made up of multiple proteins, and that's going to become important later. I've picked out one of them, complex 5 here, which I'll tell you a little bit more about in a minute. And complex 5, you can see, has multiple proteins all assembled together in a complex in the membrane to do its job. Okay, so what's happening here is electrons are coming from the food molecules, and if you follow the little red balls, they are flowing through from one molecule to another, and ultimately to oxygen, and that converts oxygen to water. And coupled to that, some of these protein complexes here, 2 and 3 and 4, are involved in passing the electrons along, and complexes 3 and 4 and complex 1 over here, coupled to that movement of electrons, are acting as little pumps, and they are pumping protons, hydrogen ions, which are positively charged out through the membrane. So what is happening is some of the energy being released by this flow of electrons is being captured in this charge gradient, all these positive charges accumulating out here like a little battery. So the membrane, the inner membrane of the mitochondria, has built up a charge gradient, storing some of that energy like a battery. Now, that energy can be released and used for a variety of purposes. One of the purposes, but only one, is to make ATP. And that happens when protons come back through complex 5 with their positive charges, and coupled to that, there's a little motor here that spins, and as it spins around, it makes ATP, which is a sort of a transportable form of energy in the cell that can be provided to the rest of the cell. So how are we going to measure this? Well, what we do is we measure the rate of oxygen consumption here, because every time these electrons arrive here at oxygen, they convert it to water. That's why we consume oxygen when we carry out respiration. So you can measure the rate at which this is all happening by measuring the rate of consumption of oxygen. And the equipment that I'm going to talk about in a minute essentially measures that. And we can then ask the question, well, what does each of these complexes contribute to this process? And we can do that because if we inhibit one of these complexes, the rate of oxygen consumption will drop. And the size of that drop tells you how much that complex was doing in respiration. For example, if we inhibit complex 5, the protons can't come back in here, and that means not as much oxygen gets consumed over here to create the proton gradient to support that. So that drop in oxygen consumption, in the case of inhibiting complex 5, will tell us how much complex 5 is actually doing in intact living cells. So that's what we're doing, okay? Now, along the way, some of the electrons leak off. Instead of going the way they're supposed to go, they go straight to oxygen first. And if they do that, they generate toxic intermediate forms of oxygen that are really, really reactive. So they're called, imaginatively, reactive oxygen species. And they are so reactive, they will react with and damage any component in the cell. And they are often used as an indicator of problems in the mitochondrial membrane. Okay, now, what do we think is going on in ME-CFS? So this is a little model that I'm going to show you first so you can see the conclusion that we've come to before I actually show the data that led to that conclusion. So what we have here is a cartoon on the left side of the healthy normal state of the inner mitochondrial membrane, and on the right-hand side what's going on in the lymphoblasts in these cells from ME-CFS patients. And there are a number of key things that you will notice. Firstly, I've got a little thin arrow here for ATP production instead of a thick one. That is to signify the fact that complex 5 in ME-CFS patients is working inefficiently. It is less able to make ATP efficiently. The second thing that's going on is that in an attempt by the cell to compensate for that inefficiency, it turns up, it upregulates its expression, its creation of all of this machinery in the mitochondrial membrane. So it upregulates the production of these proteins here that I haven't talked about so far, that are in the mitochondrial membrane and are necessary to import and export molecules across the mitochondrial membrane, exchanging them with the rest of the cell. For example, food molecule intermediates that need to be oxidized in the mitochondria, they have to be brought into the mitochondria first. And those processes cost some energy. They cost some of that charge gradient in the battery that we talked about just before. They are all elevated, as you'll see later. The other thing that the cell does is it upregulates its production of all of these respiratory complexes so that it's trying to cope with the fact that its mitochondrial ATP production is inefficient. And we'll talk more about that and the pathways that might be involved in doing that later on. The end result is that at rest, the cell is able to bring its ATP productions back to normal even though it is inefficient because it's got more of all the machinery to make the ATP. And as a consequence of that, at rest, the cell looks fine. But because in order to achieve this, it had to turn everything up, its capacity to do more, should there be an additional acute energy demand on the cell, is not there. It's already, it's like a car operating on 3 cylinders instead of 4 or 5 cylinders instead of 6. You put fuel in the tank and it doesn't make any difference. You're already getting the best you can get out of that particular car, okay? So that's the basic idea, and I'm going to show you now the evidence for it. So at this point, I'm going to ask the camera guys to focus only on me and not on the data. So my way of trying to protect our ability to publish our data is that hopefully you won't get to see the data from this point on. I apologise to the people who are looking at this live streaming because you're going to have to focus on me instead of the data in the future for the next part of the talk. All right. Oh, actually, sorry, I jumped the gun. You can go back to the slide. My apologies, I forgot I had to do this. So this is just to illustrate to you how this works. So you put your cells into this machine called a seahorse extracellular flux analyzer that Cara will tell you more about later. You measure the basal rate of oxygen consumption. You add an inhibitor of complex 5, which is the one that makes ATP. The oxygen consumption drops. That drop tells you how much ATP is how fast ATP is being made in the cell. You then add another drug, which is called an uncoupling agent or an ionophore, and what it does is it makes that membrane permeable to protons. So remember, in the diagrams that I showed you and in the cartoon and the animation, the mitochondria are pumping protons out all the time to create that charge difference across the membrane to store energy in the battery. what this does is it short circuits the battery. So the protons, as they come out, as they get pumped out, just come straight back in through a short circuit. And so the whole system works as hard as it possibly can, trying to pump more protons out, and they just come straight back in. So you get a measure there of the maximum capacity that the system has for consuming oxygen. That's therefore a measure of the maximum rate at which those mitochondrial complexes can actually operate. You then add an inhibitor of complex 1, and you can see how much complex 1 is contributing to that. At that point, then, there's only one complex now starting the process off, and that's complex 2. So you add another inhibitor, you get another drop that tells you how much complex 2 is doing. And then finally, down the bottom, after you've added all the inhibitors, you've blocked all mitochondrial activity and respiration, there's still some oxygen consumption. And that is because there are other enzymes in the cell that consume oxygen directly. So this can act as a sort of a surrogate measure of metabolism overall. So that's in the mitochondrial tests, and then you can also, in the same machine, because it's a clever machine, you can also separately, in separate experiments, measure the rate of glycolysis, which is the pathway that provides food molecules from carbohydrates to the mitochondria. And one of the byproducts of that pathway working is that it acidifies the medium that the cells are in. If you set the experiment up the right way, you can measure the rate of acidification, the rate of change of the pH, and that tells you, so this stands for extracellular acidification rate, that tells you how fast that glycolytic pathway is running. It's important because in the literature there has been suggestion that maybe glycolysis is impaired in these cells. I'll show you in a minute that's not. Then you can add an inhibitor of ATP synthase, and that means that now ATP synthase is not being used anymore, so glycolysis is no longer able to keep providing food molecules. Instead, you get maximum capacity of the acidification rate, so it jumps up. That tells you what the maximum rate is that the glycolysis pathway is capable of, and then you can add an inhibitor that inhibits the first enzyme in glycolysis, and the drop and the difference there tells you how much glycolytic capacity the cell has in reserve. None of those things are changed. Okay, now we can focus on me instead of the slides. So, first of all, what things are unchanged? The talk has four parts in terms of the results. The things that are unchanged, the things that are changed, and then we've looked at all the proteins in cells, the so-called proteome, to see what they actually do. So, what is unchanged in these lymphoblasts when you compare CFS and control is the mitochondrial mass. That's really a measure of how much mitochondrial membrane is in the cell. That's no different between CFS and controls. Also, the number of genome copies as measured by the number of copies of particular mitochondrial genes. Mitochondria have their own little tiny genome inside them with 13 protein coding genes in them, and that's not changed either. So that confirms that there's no significant difference in the amount and mass of mitochondria in these cells. So the changes that we are seeing are not because of changes in the amount and mass of mitochondria in the cells. The glycolysis rates are unchanged, the glycolytic capacity is unchanged, the glycolytic reserve is unchanged. Glycolysis is operating at the normal rate in these cells. ATP synthesis rates, after the cell has adjusted to the circumstance it is in, are also unchanged. So you can see down here, this is just a measure, a snapshot of the steady state level of ATP in the cell, and that's unchanged. And over here is the rate at which the mitochondria are making ATP, and that is unchanged. And in addition, the rate at which those toxic reactive oxygen species are generated is unchanged in the cells. Some of these results are not expected from what we've read in the literature, but that's what happened. Okay, so those are the things that are unchanged, and they actually fit with the cartoon model that I told you about earlier. What are the things that are actually changed in the mitochondria? Well, the first thing is that, as I said earlier, complex 5 is operating less efficiently. But the basal oxygen consumption rate is is not actually very different. So here's the basal oxygen consumption rate. There's a slight increase here in CFS compared to controls, but it's not statistically different. So the cells have brought their basal activity of their mitochondria at rest back to normal by their adjustments that they've made. But their ATP synthesis rates as a percentage of that is significantly lower. It's about 25% lower on average. Some patients it's greater reduction, others it's a smaller reduction. The proton leak is also dramatically changed, and the non-mitochondrial oxygen consumption is also dramatically changed, and they are elevated. The proton leak is a euphemism for all of the things that charge gradient across the membrane might be used for in the mitochondria other than making ATP. Okay? So one example is transporting protons into the mitochondria. That's using that proton gradient to drive as the energy source without using ATP. And so it's consuming some of that. So people talk about it as a proton leak. It is not a leak. It is actually the consumption of that charge gradient to do other things other than make ATP. Okay, and that is dramatically elevated, and that's consistent with the model that I showed you where all of the proteins that are importing things into the mitochondria are upregulated. There's more of them, and they're importing more food molecules to try to cope with their new circumstances because the complex 5 is inefficient. And the non-mitochondrial oxygen consumption rate is also elevated in the cell, in the lymphoblasts, and that means that metabolism in general in the CFS lymphoblasts is operating at a faster rate, and those non-mitochondrial enzymes that consume oxygen are operating at a faster rate, consuming more oxygen. Okay. Because the mitochondria now have a greater capacity through the first 4 complexes in the membrane, the first 4 respiratory complexes to pump protons out, they've got more of complex 1, 2, 3, 4, and 5, and they're trying to make up for the inefficiency of complex 5, so they're pumping more protons. That means that their mitochondrial respiratory capacity is increased. So you can see here, when you add that chemical that tells you what the maximum capacity of the mitochondria is, it is elevated in CFS. You also see an elevated membrane potential in the mitochondria, and that's because more protons are being pumped out, but that extra capacity can't be used because ATP synthase, complex 5, is not working as efficiently. So it doesn't use as much of that extra capacity that it has. And complex 1, which is the main thing, as you saw at the beginning, is the main complex that's generating this charge gradient across the cell membranes. It's also doing more in these cells, or it's capable of doing more were that being used fully by the cells. Now, so those are the things that go wrong in the mitochondrial respiratory electron transport chain. How do they correlate with the patient's experience? So we used Alice Richardson and Brett Lidbury's weighted standing time as a measure of disease severity. And you can see there's a significant correlation in each of these measures in the lymphoblasts that we make and study in culture with the severity of the disease. So on the left-hand side is more severe, and the right-hand side is less severe, and the controls are all little pink squares that you can see there. So ATP synthesis efficiency is lowest in the patients that have the most severe disease. The proton leak is greatest in the patients that have the most severe disease. The maximum capacity of the mitochondria is greatest in patients with the most severe disease. Complex 1 activity is greatest in patients with most severe disease. And that non-mitochondrial oxygen consumption is greatest in patients with the most severe disease. So, this raised the question, well, can we show that in fact there is more of these respiratory complexes in the mitochondrial membrane? So we used an assay where you use antibodies that are raised specifically against particular proteins in those complexes, and you can use those antibodies in a technique called Western blotting. The name doesn't matter, even though it has an amusing history. You use the reaction of that antibody with the protein to measure how much of the protein is there in the cells. And when we did that, we found that complex 1, there's more of this particular subunit in the ME/CFS lymphoblasts. Complex 2, the same thing applied. Complex 3 and 4 were also elevated, but not significantly so in this test with the sample size that we had. But everything is consistent with an elevated capacity of the cell to do more in respiration as an attempt to cope with inefficient complex 5. To look at that more closely, we then decide we will look at the proteome of the cells. So what you do here is you basically make an extract of the cells and isolate all the proteins. You break the proteins up into little bits and you send them through this fancy schmancy machine. And the fancy schmancy machine gives you results back that tells you what the identity of the proteins was and how much of them there were. And it's called proteomics. And it's not able to actually detect every protein in cells, but we were able to detect 3,700 proteins in these lymphoblasts using 23 patient cell lines and 16 of control cell lines. 80 of the proteins were significantly downregulated, significantly less of them in the ME/CFS cells. And when you take the names of those proteins and you can put them into an online site that sort of has a database of all the interactions between proteins, and it'll draw the interactions as networks. So each line between each of these little circles represents a link, an interaction between two proteins. And you can see there are 3 main clusters, and they're mostly to do with aspects of cell growth and division, and we don't need to go into the details, okay? So there's 80 that are significantly downregulated. There's 137 proteins significantly upregulated. And we got more clusters here when we did the same thing, looking for clusters of proteins with related functions that interact with each other. But two of those clusters stood out of interest. One were mitochondrial respiratory complex assembly and structure. That's this cluster. And the other one was a set of proteins that are also mitochondrial proteins involved in metabolizing lipids. And you're going to hear from other people here about metabolizing lipids in these cells. So we focused again on the mitochondrial ones and asked the question, is this significant? So we went back and asked it in a different way. If we take all proteins and so here's our total of all the proteins, and here are the ones that are, here's a set of proteins that are upregulated, and here's a set of proteins that are downregulated, the numbers, this time setting a different cutoff for the significance. And you can see that 1.34% of all the proteins in the cell were upregulated and 0.91% were downregulated, so less of them in ME/CFS compared to 3,650 odd that were not significantly different between the two sets. If you take out of that all the mitochondrial proteins, you can detect 311 mitochondrial proteins. To give you a feeling for it, there's probably 1,500 of them actually, so we aren't detecting everything. 9 of those were upregulated, none were downregulated. And you can ask the question, is that difference, 9 out of 302 versus 50 out of 3,700, is that significant? And the answer is yes. It's a significant difference. So that means that mitochondrial proteins are enriched amongst the upregulated ones and depleted amongst the downregulated ones. Over here, if you then ask the question, all right, let's look at those 311 and let's say, okay, I'm going to ask how many of them look like they're going up even if it's not statistically significant. Let's say how many of them have an increase of more than 10% over what you have in the controls, and how many of them have a decrease in levels of more than 10% compared to controls. And in the mitochondrial proteins, 180 increased by more than 10%, 80 decreased by more than 10%, and that difference in proportions is statistically significant. If there was no upregulation of mitochondrial proteins, you'd have expected even numbers on the two sides. Okay? So that says mitochondrial proteins are upregulated. Focus in on the respiratory complexes, which I talked about before. Complex 1 here, significantly upregulated. There are 44 subunits in complex 1. We detected 31 of them. 23 out of 31 were upregulated, and that was significantly different in a binomial test, which asks the question, if I were to toss a coin, and how many heads would I get, right? So would I get 23 heads out of 31? If I did, I'd think I've got a biased coin toss, right? So that's what that means. And we can also test the actual extent of the upregulation. So on average, about 56% higher levels in the ME/CFS cells. Complex 5 was the same down here, on average about 13% higher. And in this test, complexes 2, 3 and 4 were also upregulated, but not statistically significantly. Okay. And this just is more of the same, basically just showing you the same thing for specific complexes. This is complex 5 here. 3 were upregulated, none were downregulated in the cells. Okay. Complex 2 subunits, we found the same thing. Now, the problem with complex 2 is complex 2 only has 4 subunits. So if you think of 4 coin tosses, you've got actually a pretty good chance of getting 3 out of 4 being heads, right? And so we couldn't say that this was statistically significant, even though every one of them, there were higher levels in CFS than there were in controls. So it's not statistically significant, but it's sort of part of the emerging pattern overall. And remember, the other method that we used with the antibodies did show complex 2 was significantly upregulated, significantly higher levels. Okay, what else? So the fatty acid degradation that I talked about earlier, you can do the same thing. You can pull out all of the enzymes that are involved in using fatty acids as your energy source instead of carbohydrates like glucose. And the reason that matters is for an ME/CFS cell making ATP inefficiently, it can get more ATP per carbon that it burns, that it oxidizes, out of a fatty acid than it can out of a carbohydrate. And so it can upregulate that in an attempt to get back to normal levels of efficiency. And if you look at the enzymes involved in mitochondrial beta oxidation, you can see that of the ones that we detected, 17 were upregulated, only 3 were downregulated. Mitochondrial beta oxidation, oxidizing fatty acids to get energy, feeds intermediates into another cycle called the TCA cycle, and I don't have time to go into it in any detail for you, but the enzymes in the TCA cycle were also upregulated in the mitochondria of the ME/CFS lymphoblasts compared to the controls. So for those of you who don't know what a fatty acid is, here's a prop I made earlier. So this is a fatty acid, and what happens in beta oxidation is this has a long chain, and each bend here represents 1 carbon atom in the chain. And what happens during beta oxidation is this gets broken down 2 carbon atoms at a time. So first of all, these last couple are cut off, and then they get fed into the TCA cycle. And then the next couple get cut off and fed into the TCA cycle. And then the next couple get cut off and fed into the TCA cycle. And a consequence of that is you break down the fatty acid through this cyclic process. So you start off here with your fatty acid molecule with its long chain. There's a series of reactions that sort of prepare it, and then you cut off that last 2, and the molecule that you then feed into the TCA cycle for getting energy is called acetyl-CoA. Think of acetic acid and vinegar, it's sort of related to that. It's got 2 carbons in it. So that's one of those little 2 carbon units that goes into the TCA cycle over here, and here's where you then oxidize that and then feed the results into the mitochondria. Okay, so those cycles are also upregulated in the cells. To do that to fatty acids, the cell has to import them. And I mentioned before that the proteins that import things into the mitochondria are elevated and upregulated as well. Here's the one that imports fatty acids, so ignore all the other stuff around it. But the important thing is that there are these 3 proteins here that are involved in bringing fatty acids into the mitochondria. There's more of them as well, as you can see. One of them, on its own, just the increase in levels, was statistically significant in the proteomics. The other two also followed the same pattern but didn't reach statistical significance. So they bring the fatty acid in, and then it goes through beta oxidation, and that acetyl-CoA gets fed into the TCA cycle, also called the Krebs cycle. And here's the key enzymes involved in beta oxidation, each of them, and you can see each of them is upregulated in the mitochondria, not individually significantly so, but the fact that all 6 of them are up compared to the controls, again by the binomial test, toss a coin, the chances of getting no heads out of 6 is pretty low, and so that's statistically significant as well. Okay, what is upregulating this? Why are the mitochondria doing this? The reason is that the cells are under stress. They're under energy stress because they can't make enough ATP efficiently, and so they have stress-sensing proteins in the cell that are able to sense this problem and then in turn try to upregulate all of these mitochondrial proteins. There are 2 main ones called TOR complex 1 and AMPK. TOR complex 1 is up here. It has 3 main subunits, TOR, RAPTOR and LST8. For the listeners elsewhere, the names don't really matter that much. And then there's another protein called AMPK. We are embarking on studies to measure the levels and activities of both of these, but we've done it already for TOR complex 1, and I'll show you the results down here. So you can see that TOR complex 1 activity in the cells is elevated, and that could be the reason why the mitochondrial function is upregulated and the mitochondria are increasing their levels. And that also correlates with disease severity. Not a brilliant correlation, but it's there. Okay, so we think that what's going on is that molecules like AMPK, proteins like AMPK and TOR complex 1, stress-sensing proteins in the ME/CFS cells are sensing the problem that the cell has. They're trying to deal with it by upregulating what's going on, and they are doing that by upregulating mitochondrial function. But complex 5 has a problem, so they can bring their ATP production levels back to normal by doing that, but they can't go beyond that. So that if additional acute energy demands are placed on the cell can't meet them. So that then explains why there is this, well, it's a sort of a cellular equivalent, if you like, of chronic fatigue. So just to show you how complicated this is as a final take-home message, this is TOR complex 1 here. These are 3 of the other proteins that interact. So there's the TOR protein itself and 5 interactors. You can ask the question, well, what is known about other proteins that might interact with them? So here is the network with 10 interactors. You can keep going. What if I ask for 50 interactors? There's 50 of them, all interacting with TOR complex 1. What if I asked for indirect interactions between those proteins? And then you get a whole heap more. And for every one of these proteins, you could do the same thing. So in the cell, you have a complicated network of interacting proteins that are all talking to each other and listening to each other and regulating each other and interacting with each other. And what is going on is that there's something wrong in one part of this network, and that is then in turn causing cascades of effects through the whole network. I'm putting this up to give the non-scientists among you a sense of the complexity of this problem in the cells. It's not something that is going to have a single simple cause that you can solve with a single simple manipulation. We have to understand what is going on in these networks. So the conclusions are ATP synthesis by complex 5 is less efficient, non-mitochondrial oxygen consumption and mitochondrial respiratory capacity are elevated, respiratory abnormalities correlate with disease severity, the proteins involved in transporting molecules across the membrane and in mitochondrial fatty acid are upregulated, TOR complex 1 activity is elevated. As a consequence, ME/CFS cells may be less able to respond to additional acute energy demands. but the cause-effect relationships remain to be determined. And that's my last point. When you have a network like that, I've given you one scenario where we start out with a complex 5 defect and we can explain in a model why everything else is doing what it's doing. But it could be the other way around. It could be that there is a leaky battery, and that's why there's less proton gradient available, less charge gradient available for ATP synthase to operate. So the defect in complex 5 could be secondary to some of the other things that are going on in the cell. To dissect cause-effect relationships, you need more than correlations. You need to manipulate one thing at a time in the cells and ask if that can cause all the others. And that's what we're doing next. Thanks.
In the interest of time, 2 questions. I'll open the floor. Yes.
Well, thank you. That was very elegant. Just 2 questions. How many CFS patients did you take your lymphoblast from?
So at the moment, our CFS cohort is 57 patients, and unfortunately fewer controls. We've only got 17 controls at the moment. We're trying to build that up. We want to get to 100.
When we ran the seahorse trials across our patients, we found different problems in different parts of the cytochromes. Have you found anything apart from complex 5?
No. So when we look at the efficiency of complexes 1 to 4 and they're expressed as a percentage of the maximum capacity, that's the same. We aren't measuring cytochromes directly though, so we're just measuring the activity of complexes 1 to 4.
So could it be across the world there could be many different causes of cytochrome damage which would be different in each place with different infections or different things but similar outcomes?
I think all bets are off. I think anything is possible as primary causes here at this stage.
Yes, Matt. Yes.
Could you say whether the uncoupling proteins were among the proteins that were upregulated?
No, I looked for that, but unfortunately the uncoupling proteins were not amongst the ones that were detected in the proteomics.
Can I ask you for how long your patients were ill with ME/CFS? The underlying question I have is whether you think this is an early problem in ME/CFS, but it's only a problem that develops after some time with consequences.
I'll have to ask my collaborator Brett Lidbury to answer that question because I'm unaware of the answer.
It was quite a range, Louis, probably just from a couple of years right through. I think we've seen with Don Lewis's cohorts up to 30 to 40 years sick. But we didn't just have extreme cohorts, different lengths of illness.
I think in the interest of time and to keep the schedule on track for this morning and then flowing on for the rest of the day, if anyone has any further questions at morning tea, perhaps that would be advantageous to seek out Paul and ask those questions. I, for one, would like to ask a question at morning tea. I'll now hand over to my colleague, Professor Donald Staines, and he will be introducing the second speaker of this morning's session. Thank you.